We change gear now a little bit with our next talk. It's quite exciting to have Dr. Adam Gerstenecker from the University of Alabama at Birmingham here on stage. He actually took a closer look at how we evaluate PSP symptoms in the clinics. Thank you.
Hi, thanks for having me. I'm actually going to be on top of it here with the time, so you might not have to do that. So my talk today is going to be about behavioral, cognitive, and functional aspects of PSP. And I don't have any relevant disclosures. I have more slides than I can get through, so I'm going to hit a lot of the high points here, but I am under the understanding that these can be found later, so if people want more in-depth, please go look at it. When I talk about cognition in PSP, one of the things you find is that it's primarily a subcortical dementia. And what's meant by that? So the number one impairment in PSP when we speak of cognition is something called executive dysfunction. And executive dysfunction is associated with the frontal lobe, and it regulates things like multitasking, switching from topic to topic, planning, organizing, things like that. There's also slowed information processing. There's limited involvement of memory and language, but we do find some memory and language impairments. So if we look at this slide here, if you look at the top box, that's a score of what's called general cognitive impairment. Now, those numbers there are how many people are impaired. The first number, or the first column, the 42.6, is at the 5th percentile. So that means that these people were doing worse than approximately 95% of the population. Impaired at the 1st percentile is doing worse than 99% of the population, and this is controlled for age and education as well. So it's far more than what you would see in a normal population. If you jump to the second red box and the third red box, those are measures of executive dysfunction. As you can see, the numbers jump up, so you can see that the primary driving force behind this general cognitive function loss is loss in executive dysfunction. If we jump to total number of tests impaired, as you can see, only 0 people, when we give approximately 4 or 5 tests, only 0 people, or only 24% of people showed no impairment. However, about 17% of people showed impairment on 3 or more of the tests we gave, so almost every test that we gave. Conclusions from the cognitive study was that, like we said, executive dysfunction is the primary impairment in cognition in people with PSP, and we do think that cognition should be better assessed in patients with PSP in the clinic. Normally they're getting the MMSE, but everyone who took our test scored greater than 24 on the MMSE, so they were being cleared cognitively in the clinic, and then they come to a neuropsychologist and we find pervasive deficits. I'm now going to switch gears and talk a little bit about behavioral abnormalities in PSP. This is more along what you would find in a psychiatric clinic. This is the chart. We gave something called the Neuropsychiatric Inventory, and all of these domains on the left are what the Neuropsychiatric Inventory assesses. This is a caregiver report, so it's people close to the patient who are filling this out. The primary thing that we found was apathy or indifference. I'm going to talk a little bit more about apathy in a couple of slides. We also found quite a bit of depression. Depression in our study was more elevated than we had seen before. But in the paper, we think it was accurate, and I'll talk a little bit about that as well. As has already been discussed at the conference here, sleep and nighttime behaviors were very elevated. Also eating behaviors. And then if you look at the next group, we have elevated agitation, elevated irritability, elevated disinhibition. So it's a hard thing to manage from a caregiver's perspective, and we recognize that. There weren't a lot of correlates with these things. Disease severity were not correlated. Disease duration was not correlated with these things. So behavioral abnormalities will hit anyone at any stage in the disorder. So one of the things we find, and one of the things we find when we talk to caregivers of PSP, is dealing with apathy. So apathy is not caring, someone having kind of a blasé attitude about life. It's not depression, it's more just not caring than feeling sad. For families, many of the families will believe that the patient is not interested because of something they're doing. But what is important to understand is that a lot of this is because of the underlying neurological deficits is leading to apathy, and it has nothing to do with the caregivers. Apathy and depression, like I said, a lot of times these two syndromes are confused in the clinic. So a lot of people with PSP are being prescribed antidepressant medications when they don't need to be, because it's actually apathy. It takes an in-depth look to be able to untangle apathy and depression. But when we looked at the two with the NPI, we found that they were not statistically correlated and shared only 1% of variance between the two. So it didn't look like they were related with each other at all. However, we did find a lot of depression, and one of the things we called for in the paper was for more studies on depression in PSP. There aren't a lot of studies about cognitive behavioral therapy in PSP, if therapy works in general, how antidepressants are doing in PSP, because so much of the samples are filled with people with apathy. We also think that since these syndromes were distinct, that the depression we see in PSP is likely not a function of the underlying neuropathology, but it's probably a function of what the disease is doing to the person. So again, things like therapy might be able to help, behavioral activation, things like that. I'm going to turn now and talk a little bit about functional ability. We talk a lot of cognition in PSP. There's a lot of papers on cognition in PSP, but to date there's absolutely 0 papers on functional ability in PSP. So how the cognition is impacting the person at home, how it is impacting the person in the way they live their lives. There's two, in the clinic, from a clinical standpoint, there's 2 types of functional abilities or daily living abilities. One is basic ADLs, and that's like bathing, dressing, grooming. And another one, which is the one I'm going to focus more on, are IADLs. That's more highly correlated with cognition, but is also very crucial to independence in the home, like financial management, how well a person knows about the treatments that are going on or can manage their medications. So functional ability is not only important to know about what's going on in the daily lives of people, but it's also important for people like neuropsychologists and other clinicians when they're talking about mild cognitive impairment and dementia. So mild cognitive impairment is when the person has cognitive impairment but no or minimal deficits because of cognition to functional ability. When you transition into dementia, the cognitive impairment has begun to erode on the ability of the person to live their daily lives. So normally functional ability in PSP is assessed with some of the disease severity scales, but we gave performance-based scales to people, and this means we actually had them do the tasks in the clinic. So we gave them scenarios about treatments and asked them to tell us what was going on in them. We asked them to pick up a phone and call 911, and we also asked them to do a number of financial tasks. Overall, we found that over 80% of the sample had significant declines in functional ability. Primarily, this was in medical decision making, interestingly enough. So the people that we talked to did not show the ability to truly understand what was going on around them medically. They needed a lot of help, a lot of cueing, more than they're getting in the clinic. We also found that financial decision-making was poor, and one of the scary things that we found from a caregiver perspective is that the sample was especially susceptible to scams, so they would be willing to give their money to people who had dubious intent. Although it wasn't as impaired as the other two, we found that over 50% of our sample did not have the ability to pick up the phone, dial 911, tell the dispatch operator what was going on and where to go. So that's also something to keep in mind. And interestingly enough, we also found that caregivers were not good at quantifying the level of IADL functioning in the patient. We found that they thought they were doing better. So the understanding there was not so great. They didn't understand that the people weren't understanding what's going on around them, that they might be at particular risk to give their money to people who they shouldn't give it to, that they're having trouble even dialing 911 and things like that. And with that, my time is up, and I've had a lot of people help me out in the business, and I'd like to thank them, and I'd like to thank Cure PSP, NIH, NIA, and NDS, who we've all had funding through. Thank you very much.
the idea to keep the questions, this came up during the last session because I really enjoyed listening to your questions, which are really important because sometimes, my background is also neuroscience, and sometimes you just get a bit blindfolded and you don't really understand what the real needs are and where we need to educate. And so I thought that was really, really valuable for us. So, first, yeah. Let's start with Ho. I always wanted to be a garbage collector when I was young because I thought it was really cool. Growing up in Germany, I thought it was a cool thing, like these orange jackets are always out, big trucks. So is there actually a natural way of boosting your defense mechanism, your autophagy, your garbage collectors? Is there any food or anything I can do?
Right. So we have tested. So we have tested it for early kind of garbage clearance. What we call like creating these garbage trucks. Something called trehalose, which is a sugar. But you have to take it in very high amounts. And as I explained in terms of some of the biology in the cell, kind of creating more garbage trucks isn't necessarily going to benefit later on in the disease stage. So that actually might be harmful depending on how far the disease has progressed. So while we can show it in cells and animals, it's probably not recommended for patients to use a natural product like that.
Right, okay.
Yeah. Sorry? Well, I'm finished the answer, so.
So, and then actually I have another question. So in your lab, are you now actually really trying to discover some sort of pill you take once a day and that could sort of, for a long run, kind of increase, and then in parallel to whatever therapy is on top of that, may actually help to remove existing misfolded tau, the garbage?
Right. So the ultimate goal really is to kind of create that drug that an individual can take to help remove garbage, be it tau or other proteins that are involved in neurodegenerative diseases. Some of the wicked compounds that I discussed are some of the early stages in what we call drug discovery for that pill, hopefully in the future.
Thank you. Some questions for Dr. Schellenberg. So in general, could you just maybe, because there are a couple of questions on that, could you just comment again on the risk of passing it on to the next generation? So if I'm diagnosed with PSP, is it likely that my children will have it?
So there are mutations that you have a 50% chance of passing it on in tau, but those are extraordinarily rare. It's highly unlikely unless you have a family history, a very dense family history of PSP or frontal temporal dementia. You probably don't have one of those mutations. So I can't give you, they're so rare I can't give you a percent how often they occur. And the H1, H2, as I said, you probably will pass it on to your kids, but they had such a low starting risk that even though it goes up sixfold, they're still at low risk for tau. So this does not get passed on generation to generation. That's why Dr. Golby's clustering in villages is extremely interesting, because there's something that pushes you. You have probably have a genetic load that puts you at higher risk, and then something else happens, and that might be metals or stuff we don't know about.
Yeah. And so there are also a couple of questions here for you, Jerry. You mentioned those 2,000 genomes you're going to analyze. Can people still participate, and who are those 2,000? Are they alive? What's the what's the deal? Is it a kind of a clinical trial? How can they participate? Is there?
Well, most of those people, the 2,000, are deceased. Those are autopsy-proven cases. PSP is a pretty solid diagnosis, so when we follow up, we will be using clinical cases, but kind of when we spend more money, we want to absolutely make sure the person has PSP, so most of those are autopsied. But we are always collecting additional volunteers for genetic studies because we can extend what we find with the sequencing with other technologies that are less expensive.
That's a comment from me now. So the core mission of the Brain Support Network and one of our programs is actually to support brain donations. And that really feeds into it. I think also a common theme you see this morning is like how we all work together. There are I mean, you're focused on your research, but you cannot live without other branches and people in this whole research world, I should say, because without brain donations, less material for the geneticist to analyze. And I have a question for Dr. Clark. So when you develop those, actually that's one of the cards here, when you develop these animal models, so in the long run you would like to test drugs, right? Have you already tested drugs in this or not?
Yes, the ultimate goal is to test drugs, and of course we would love to be able to find drugs that are off the shelf already approved by FDA. We've done some tentative work with the animal model, some obvious things because those neurons that we're removing actually produce this chemical called acetylcholine. Acetylcholine activates nicotinic receptors. And so we've tried nicotinic replacement therapy in our animals and we see absolutely no effect. But we are in the process of selecting other drug candidates, but I have no news on that front yet.
All right. And I just have to put my advocacy hat on here right now as member of staff of QPSP and also comment on this genetics project. I get quite a few phone calls of people who are desperate and want to be tested. What I can only recommend is that you contact experts and let you guide you or call us. And there is actually an association of the National Genetic Counseling. We strongly recommend not to go to any for-profit providers because you will receive a letter, and in this letter you get a long list of potential mutations that are associated with other diseases, and that's it. I would freak out, right? And if I would read some of those, because some of these mutations mean a bit more to me than maybe to you, but I still would pick up the phone and say, Jerry, help me, right? So, and then if that's not happening, right, there's no reason to just kind of sell all your stuff and go gamble in Las Vegas, right? So it's important, and it's actually really irresponsible. It's absolutely irresponsible of for-profit companies to offer genetic sequencing without genetic counseling. And I don't care about online genetic counseling. It needs to be face-to-face and direct questions. And the big centers, I know that Columbia University does it. I'm sure that in Buffalo there's also a big gene center. They all offer it, and if you go to the website of the Genetic Counselors Association, I think it's called, they provide a finder where you can type in your zip code and get an analysis. But as we heard from Dr. Schallenberg, it's not so important anyway to get tested for you privately. For research, it's really important and will give us a lot of information. So let me just scan through. We have done, most of them are actually quite the same. Let's see if I have another question. Beg your pardon? I didn't hear it.
Nothing wrong with ending early.
Ending early, all right. But I have another question for Dr. Clark, and is also, or more, it's Exactly. It's actually more like a comment also, because, you might wonder, Dr. Clark's work, where is that? Does it help you right now? Well, maybe not directly, and you can actually directly, as in we have a drug tomorrow, and you can certainly comment on that. But it's really important that we have also this preclinical work, because if you go back in time and look at the last 4 decades, let's say, about neurodegeneration clinical trials, it's quite of a sad result so far, right? And we need better animal models. And my background is actually, I'm a behavioral neurobiologist, so I really wholeheartedly support also what seems to be basic neuroscience is really important for the future, and we need to invest in that as well. So if you just want to say a few words.
My only comment is you've seen a lot of data in cells, in other model systems, and I just want to impart on you my feeling of the importance of the animal research. Our brains are very complicated organs, and without using the whole organ system, these questions that we were trying to unravel, we will not be able to do it in a dish. And so, personally, after I finished my PhD, I wanted to leave animal research behind and not ever do it again. But as our understanding of the brain evolves and grows, it's really important to go back to it. So I just want to emphasize my wholehearted, deep appreciation for the animals that we use, and that it's very valuable that we support animal research for this complicated organ that we're trying to fix.
Well, thank you. That's actually exactly how also I felt, using animal models to work in the lab and to find a cure. So first of all, I would like to thank all speakers. We had to cut this a little bit short. I'm sorry. It was an absolute pleasure to be here today to talk to you. I hope you could learn a little bit and take some stuff home with you. Maybe if it's even more questions, we keep them. I think it's really valuable what you were asking. I would like to go through this. We have a little booth in the back. This is Cure PSP. There are also some of my business cards are there. I unfortunately have to leave because Cure PSP has its board meeting right now. But my colleague Sabrina in the back, she can help you. And we will continue, this afternoon to work on how we can actually make this foundation better and serve you, and continue the beautiful collaboration with Robin Riddle. Also, on those business cards, there is my telephone number and my email, and as I was introduced as a translator, I'm happy to continue translating to you. If you have questions, call anytime. That's what I'm here for, and I'm really looking forward to talking to you. Thank you.